Chapter 6 focuses on nucleic acids and protein synthesis, the biological process by which new proteins in the body are synthesized. Under this chapter, we will be starting off by looking at the structure of nucleotides. Now, you have two major types of nucleotides you should know, and then apart from that, you have a sub type of nucleotide you should know. So, let me write down as a point. Now, the main two and sub 1 nucleic acids we are concerned with a semicolon. Right. The main two are going to be first something you're very well familiar with, DNA, and number two, it's going to be RNA. I will also give you the full forms of these right now. They are nothing new. You have known these names since your IGCSEs. Deoxyribonucleic acid. And this does not have the deoxy part. This is simply ribonucleic acid. Ribonucleic acid. And then what's this subtype of nucleic acid you should know? So this is not really a nucleic acid itself like DNA or RNA, but it has almost all the properties of a nucleic acid. That's why we include this here in somewhat of a manner. Now, when it comes to Cambridge Biology 9700 specifically, you will be touching up on this molecule in much more detail under A2 levels. The starting chapter, chapter 12, would be energy and respiration. Under that, you'll be talking about this molecule in a lot more detail. As for AS levels, which this course is currently pursuing, you don't have to know about this molecule that much, just its basic structure. Now, if you have not really guessed already from all this information what this molecule is, we are talking about ATP, the universal energy currency. In biological systems and biological states, ATP would almost always be the energy carrier. Now, what's the full form of ATP? ATP stands for adenosine triphosphate. Triphosphate. When I talk about the structure, I'll tell you the small interesting points I find about ATP. Now let's move on. And okay, let me give you one more point. Now all these nucleic acids, all three of them, are actually considered to be something known as macromolecules. What do you mean by macromolecules? Macromolecules means giant, obviously. And they are also polymers. They are polymers. These nucleic acids are polymers of individual nucleotides. It's a very clear distinction you should get into your mind. Nucleic acids and nucleotides are not the exact same thing. Now, our subtopic was nucleotide structure. We were talking about nucleic acids here. Nucleic acids are made up from and made up of nucleotides. Just to emphasize on that a little bit, I'll also write this. Nucleotides make up nucleic acids. Alright, distinction very important. Right, nucleic acids. Now let's look at nucleotide structure and what a single nucleotide comprises of. Nucleotide. First of all, you have a pentagon in the middle. What do you mean? A pentagon? A pentagon represents a carbon chain. If I were drawing the carbons like this, that would look something like this. 1, 2, 3, 4, 5 carbons. What do you call a 5 carbon molecule? Or in other terms, what do you call a 5-carbon sugar? This is known as a pentose sugar. The first part of any nucleotide. This is a common nucleotide structure to all three. All right, do have that in your mind. This nucleotide structure is common to all three types of major and minor nucleotides we're going to be talking about. So this is the backbone of all of it, the pentose sugar, which is a 5-carbon molecule. Just for some more context, if you remember, if we had 6 carbons in our backbone, we called it a hexose, 7 carbon heptose, 5 carbon is therefore called pentose. Also, we have tetroses and trioses as well. None of this is new. It's just simply if you have 5 carbons, you call it a pentose sugar, right? What's the next part? Now, bonded to this pentose sugar is something like this. Here you have a box. This is not really a box. This is actually a base. And this is not a simple base. This is a nitrogenous base. That looks like an M. I wanted to write an N. Okay. Nitrogenous base. More on nitrogenous bases under DNA in a bit. And then on the left hand side, we have another bonding going. And that is going to connect to a circle. And that circle, let me get rid of that small part. My OCD is always taking over, I swear. Okay, not yet. Cool, right. Here what you have is something known as a phosphate group. This is your phosphate group. I will also write down the formula for phosphate. That would be PO4 minus. This is what the formula of the phosphate ion. Yep, phosphate ion like so. And this is the basic nucleotide structure. So if you look here closely, we have a pentose sugar bound to a nitrogenous base on the right hand side and a phosphate group on the left hand side. This is not hard to memorize, right? So you have to memorize this. You should be able to interpret and draw any form of nucleotide structure after you have gone through with this, right? Now let's look at how this applies to our three major types of nucleic acids. I will start off with deoxyribonucleic acid, make my way onwards to ribonucleic acid, and then finish up with our subnucleotide type, ATP. Okay, ATP. Now, when I started explaining this nucleotide structure to you, I clearly told you that all sub and major types of nucleotides are going to have the same exact structure. So to stay true to my word, I'm just going to Control C V, and I see a small unwanted part as well. I'll get rid of that. So exact same structure right here for DNA. We'll zoom in and keep it centered like this. Let me get rid of that small part. Okay. Now, this is the deal. It's the exact same structure for all three, actually. Let's multiplicate and do the exact same thing. So same structure again. Like so. The only thing is from one kind of nucleic acid onwards to the next, things change. Things in the sense, the type of sugar. Now, this is a pentose sugar. Now, all of them have a pentose sugar, okay? We're not changing the number of carbons in a pentose sugar. It's still a pentose sugar. We might change the type of pentose sugar. What does that mean? Let us take a look. Okay, still, this is the phosphate group. Nothing's different there. PO4 minus, I'll write down like that. Okay, and here, this is the nitrogenous base. This is still the pentose sugar, 5 carbons. Now, here's the thing. When it comes to this, the sugar is specifically something known as deoxyribose. Something known as deoxyribose. Hence the name deoxyribonucleic acid. We call it deoxyribonucleic acid because the sugar, the pentose sugar that we use is called deoxyribose. Okay, now if you actually look at this pentose ring in a little bit more detail, before when I was explaining this, I told you to count 1 2 3 4 5. Okay, 5 carbons pentose sugar. Yes, I mean 5 carbons pentose sugar, but if you actually go online and look for the structure of this, you'll see here we don't have any carbons, it's just an oxygen. Carbon counting begins from here, 2 3 4, and the fifth carbon is somewhere around this branch. Just as my introduction previously went on, I told you carbon, 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 just 5 carbons, just to give you some context. Okay, I wanted to mention that right now. I thought I shouldn't mention that right now, just to prevent any forms of confusion. Okay, know that there are no 5 carbons like this. 4 5, that was just to help you since you were just being introduced into a pentose sugar structure. Small disclaimer there, right? DNA, back to this, deoxyribose. So here the fifth carbon, it makes this the pentose sugar, and we are specifically using deoxyribose. And another very common feature of DNA is this. What's this? DNA is a double-stranded molecule. DNA is a double-stranded molecule. And what about the nitrogenous bases? Here's the thing. DNA has 4 specific nitrogenous bases. Number 1, it can be A. Number 2, it can be G, C, or it can be T. A stands for adenine. G stands for guanine. C stands for cytosine. The T here stands for thymine. Simple. Adenine, guanine, cytosine, thymine. These are the 4 nitrogenous bases that DNA would have. Moving onwards to RNA, same thing. It also has the nitrogenous base, the phosphate group, PO43 minus, nothing's different there. Still a 5 carbon pentose sugar. This is where the two structures differ. This is known as deoxyribonucleic acid because deoxyribose is being utilized as the pentose sugar, but when it comes to RNA, it's not deoxyribose, it's ribose. The pentose sugar is called ribose. Hence the name ribose. RNA is not double-stranded. RNA is simply a single-stranded molecule. RNA is simply a single-stranded molecule. Now, what about the nitrogenous bases? Here also we have 4 nitrogenous bases. So I'm going to copy paste things like so. Here's the only difference. RNA is not capable of consisting of thymine. RNA is not capable of having thymine. RNA has not T but something known as U. And this U stands for uracil, not uracine, okay? Don't make that mistake. It's not uracine, it's uracil. It's a very common mistake. It's uracil. It's uracil. We will be talking about the complementary versions of all of these in a lot more detail under DNA replication. Until now, all you have to know is here AGCT for DNA, AGCU for RNA. So I'm going to put a small asterisk. If that looks like an asterisk, never mind. To show the difference, right? Onwards to ATP. Now, to represent this, I'm going to have to move this molecule down. But why? That's because I'm going to have to fit in not 1, but 2 more phosphate groups. Let's come to the name of ATP, adenosine triphosphate. What is this? Adenosine triphosphate. That clearly links to three phosphate groups. Previously, DNA and RNA, they both just had one phosphate, one phosphate. But when it comes to ATP, we're having three phosphates. And here's the fun thing about ATP that I told you I would mention. This guy can break the phosphates. So if this phosphate breaks and goes away, this becomes adenosine diphosphate. If the second guy also breaks away, this becomes adenosine monophosphate. That's how ATP is actually capable of releasing its stored energy. Like I said, there's no details of that process required here at all. Now, nitrogenous base. Do you realize how I said nitrogenous base and not bases? That's because every single time for ATP, it will only be A. Choose a different color, it will only be A. Hence the name adenosine. The adenine nitrogenous base is being used. What about the sugar? Here's the deal. It's similar to RNA. We don't have deoxyribose here. It's a ribose sugar. It's a ribose sugar. A small point about nomenclature. Now, if I split the molecule from this part and I have this base, that's adenine. Now the base is adenine. If I couple that base with this sugar, I get adenosine. That's how the name is built. That part is adenosine. If I now couple the phosphates, then only the name becomes adenosine triphosphate. Why is my handwriting getting worse by a second? Adenosine triphosphate. That's how this wording, this nomenclature is actually built around adenosine triphosphate, the name. Okay? Like I said, this is the universal energy currency. The universal currency. If it's a biological organism, somehow, if it's a biological system, there's a very, very, very highly likely chance that energy is carried in the form of ATP. And this previous point I told you, ATP can vary between adenosine monophosphate, where you have just one phosphate, adenosine diphosphate, two phosphates, and then finally adenosine triphosphate, where it's full, it's concentrated, it's saturated with all three phosphates. Those are the three kinds of major and minor nucleic acids and nucleotide structures you should be aware of.